Good evening and welcome to the Kretz Public Lecture. My name is Brad Schaefer and I'm the director of the UCLA Kretz Center for California Conservation Science. This is our 13th public lecture and it's a little bit different than our normal ones. Back in 2012, when I moved to UCLA to head up the Kretz Center, I got together with a bunch of UCLA faculty and local conservation biologists to put together a vision for what the Kretz Center would be, what our goals were, what we're going to try to accomplish as an academic center. And the vision that we came up with was one that was a little bit different than the normal academic center. For sure, we wanted to have a center that was grounded in the strong academic scholarly science that UCLA is known for. But we also wanted to create a program where we accomplished and motivated and actually did science that makes a difference in the real world. And the centerpiece of that program was and is our postdoc program. These are young scientists who have just finished their PhDs and are entering into the next phase of their career path towards being conservation professionals. And so what we decided is that coming right out of the gate, every applicant who applies for a Kretz postdoc had to have both a mentor on campus that would ensure the scientific integrity of their work, and also a mentor off campus in a government agencies, a non-government organization, some group that consumes science and uses it to set policy, to set agendas, to make decisions. And every year we have hired a postdoc who we keep on for 2 years, who attempts to fulfill that dual mission. It's not easy, but there's some wonderful people out there, and we've had the privilege of hosting many of them at the Kretz Center over the last decade. So I decided, since this year is Zoom, this year is all virtual, it might be fun after a decade of this program to contact people, see who could join us, and hear what they've been up to. So I reached out to the 13 Kretz postdocs who we have funded so far, and to my amazement, every one of them said they would be thrilled to be involved in this program. So you're going to hear from all 13 of them this evening. The agenda is quite straightforward. Each speaker has 3 to 4 minutes to give you the answer to the following prompt. Conservation of biodiversity is an area of science that demands solutions. What do you see as a major problem in biodiversity conservation, and how is your work addressing that issue? That's the prompt. They have 3 to 4 minutes to answer that question with respect to their own work. There are no slides, no visuals. This is just individuals and audience telling you what they've been up to and how their program is going. Afterwards, we'll have a Q&A session followed by some breakout rooms, virtual breakout rooms, where you can get to know the speakers a little bit more and hear more about what they have to say. So we've organized these 13 speakers into 4 groups. The first is Elizabeth and Zach, who both work on butterfly conservation in Southern California. The second is Alex, Justin, Luke, and Samantha, all botanists working in different aspects of the conservation of California's flora, its plants. The third group consists of Yosha, Eric, Rachel, and John, and all of them are urban ecologists working on problems associated with urban plant and animal biodiversity. And the fourth is Gary, Jesse, and Dave, and the 3 of them all work on different aspects of vertebrate conservation biology. So that's the team and that's the agenda. Before we get started, I'd like to just say a few quick thanks. And the first is a thanks that I give every year, and it becomes only more and more heartfelt as our program goes on, and that's to Morton Kretz. Mort's generosity funds the Kretz Center. His gift is the centerpiece of our endowment that allows us year in and year out into the future to hire these young scholars, hire these postdocs, and help launch them into the next phase of their career. And Mort's generosity has been amazing. And as always, Mort, we thank you very much for that generosity. The second thank you is to the institutions off of campus at UCLA who have supported both financially and intellectually our postdocs. And those institutions include the National Park Service, one of our primary, probably our primary partner, both the Santa Monica Mountains and also this year, Yosemite National Park, both of whom are currently helping to support current Kretz postdocs. There's other institutions as well who are helping out. Besides the National Park Service, the U.S. Fish and Wildlife Service, the Natural History Museum of LA County has been a great supporter. And more recently, the California Conservation Genomics Project, another project that we run through the Kretz Center, is kicking in some financial and intellectual support. And the third thank you is to the individual faculty at UCLA who have welcomed our postdocs into their labs, provided intellectual support, sometimes provided financial support. Many different labs, many different individuals, all supportive of conservation biology, conservation science, and helping to mentor these young scholars. Victoria Sork has been a particularly important faculty member in that regard. There have been many, many others, and to all of you, I appreciate it. And of course, finally, for this event, it always takes individuals well outside of myself and my colleagues to help make an event like this happen. Colin O'Brien Lux and Alejandra Moreno from the Institute of Environment and Sustainability have been a tremendous source of support. And Gary Butcherelli, who you'll be hearing from in a few minutes, is my right-hand person in the Kretz Center, and I never get anything done without Gary's support. So thank you very much, Gary, and thank you to everybody. Okay, with that, I think we should get on to the next main part of our program, and that's going to start off with Elizabeth Huang, who's going to tell us something about butterflies and butterfly conservation, perhaps in Southern California. Thanks.
Hi, everyone. I'm Elizabeth Long. I'm joining you tonight from Virginia. I was a Kretz postdoc from 2014 until 2015. And since leaving Kretz, I have worked in the nonprofit sector and have been closely involved with the organization of biological field stations. My project while I was at Kretz was studying butterfly distribution, biodiversity in the Santa Monica Mountains, and also in the city of Los Angeles. My partner organization was the Natural History Museum of Los Angeles County, which was just an absolute joy to be able to work with that group of folks. And so thinking about what sorts of challenges we face in the world of conservation right now, one of the things that concerns me most is the decline in scientific literacy. This, I think, is probably apparent to everyone in this space. And I think that that decline of scientific literacy has really gone into overdrive, probably in the past decade or more, with folks not trusting science, not trusting the research that comes out. And there are several things that I hope that we can do to start to bring people back into the fold and start to gain some trust. One of the things that I feel strongly about is open access science. When I was the director of conservation science at Mohonk Preserve in upstate New York, one of the things I'm most proud about doing there was digitizing and making open access over 100 years of natural history data that had been stored on index cards in an office. I mean, tens of thousands of natural history observations. And so when I say open access, I mean making our science, making our publications, making our data available to anyone who wants to be able to see it. One of the other things that I'm really happy to be engaged with is community science. When I was at the museum during my Kretz postdoc, they had an entire department that was at the time called the Citizen Science Department. They've renamed it now Community Science. But the idea of engaging with just ordinary people in your community, in your area, or even just online remotely, to get them involved in the process of doing science. And that wasn't something I anticipated when I started my Kretz postdoc. It sort of came along with the Natural History Museum portion of things, but it's something that I'm really strongly engaged with now, and I do a lot of, and it's actually probably one of the most rewarding things that I get to do now. I also think that the professional and academic sector needs to more carefully and wholeheartedly embrace scientific communication. So sci-commerce, people who are really good at communicating with the public. And I don't believe that every professor should have to do SciComm, because no one can do everything. So, but people who are good at it should be encouraged to do it, and it should be valued. It should be valued in the promotion process. It should be valued. It should be valued when we're reviewing people's professional credentials and their achievements. So people who are good at doing scientific communication don't feel like it's an extra thing or kind of a soft thing that they're not going to get credit for. It's incredibly valuable. I mean, think of if I can talk about David Attenborough and just how much joy I've gotten from that. And imagine if we gave encouragement to folks who are young David Attenboroughs and really helped to train them, just what that could do to bring the public back into science and trusting scientists, not only trusting us, but also just to be engaged and amazed and delighted at the natural history, at the biodiversity that's all around them. So I think I'm running out of time. So I will turn it up to Zach McDonald.
Hi, Elizabeth. Thanks so much for that introduction. And yeah, so my name is Zach McDonald, and thank you very much to each of you for attending this awesome virtual event. So right now, I finished up my PhD at the University of Alberta here in Canada, and I'll be moving to UCLA to start a Kretz postdoc this October, working with professors Brad Schaefer and Tom Gillespie. So now most of my past research has been within the fields of landscape ecology and more recently population genomics, and has been aimed at basically understanding the interacting effects of habitat loss, habitat fragmentation, and climate change. So not only understanding how each of these processes affects biodiversity, but how these interact and really form emergent effects that are important considerations within conservation frameworks. So my approach to this was really that I was kind of split between two disparate fields. One was landscape ecology, and one was population genomics. And it wasn't until I actually attended the Kretz Conservation Genomics Workshop in 2018 that a light really went off in terms of how these two fields could be integrated into this emerging field of landscape genomics, which basically provides us with a really powerful toolkit, I think, for conservation planning of both wide-ranging species and very localized endangered species. And then so during my next 3 years at UCLA, I'll be working with both the Kretz Center and the California Conservation Genomics Project to study a variety of threatened and alpine butterfly species throughout the state. So now the basic pressing conservation issue that I'll be focusing on again are the interacting effects of habitat loss, habitat fragmentation, and changing environmental conditions. And I think this is of particular interest to the butterfly specifically because many species exist in what we call a metapopulation framework, which is just basically you have subpopulations that are distributed across the landscape, and each one of these subpopulations have their own independent population dynamics. So they'll go extinct on their own, and then maybe as individuals disperse from adjacent subpopulations, you'll be able to get a new population going again. And then a really important thing that goes on in these metapopulation networks is gene flow. And then so that's really something that we're interested in, is that as these populations become smaller and more isolated, both because of like habitat loss and the fragmentation of them and the changing environmental conditions at the local scale, we're interested how is movement between all these populations and the movement of genes across the landscape going to change. And then so to really model this and to understand it, I think we'll require equal parts population genomics to understand how genes are moving across the landscape, and forward in time landscape and environmental modeling. And then so this is why I'm going to be working with both Brad Schaefer, who has a really strong population genomics background, and also Professor Tom Gillespie, who has an incredible background in this landscape and environmental approaches, to really basically predict in the future, how is the relative configuration of habitat on the landscape going to change. And then so I believe that butterflies, among other groups, will require basically translocation efforts where we're moving individuals across the landscape, where we're physically intervening. And we're not only trying to move these individuals to facilitate gene flow on the landscape, but we also might want to move individuals, say, more north, actually expanding their range to track these changing environmental conditions, or maybe we're moving them up mountain slopes so that they can persist into the future. So I think that's a good place to leave off. And with that, I'll pass this off to Alex, who's gonna, I think, tell us about some pretty cool plant stuff. So thanks for that.
Hey, great. Thanks for having me. So I was a Kretz postdoc from 2015 to 2017. And I worked with Bill Rundle, John Keeley, Marty Witter with the National Park Service, and also Uli Zeit. And when I was at UCLA, I studied plant drought responses in the Santa Monica Mountains, and especially how those traits are associated with flammability and fire danger. And this was really cool because my work contributed to LA County Fire recently amending the threshold of fire danger that they use. And now I'm currently a postdoc at the Pacific Northwest National Lab, and I'm working on a project called Next Generation Ecosystem Experiments Tropics. And my current project is this decade-long Department of Energy funded project where the overall goal is to improve our understanding of how climate change will impact tropical forests by the end of the century. Now, this is really important because tropical forests play a very important role in global carbon and water cycles. Now, pre-COVID, I got to travel to tropical forests all around the world, collecting this empirical data in order to inform and parameterize models. But now I've been able to actually implement computer modeling in order to understand and predict how tropical forests are responding to drought. So my work is a little bit different, working in the tropics rather than in California, but overall, I think that climate change is really the greatest threat that we face, whether it's in California or any other system. And so its impacts is, this is both a scientific and societal problem. So I think that is going to be this continuous thread throughout my career, regardless of where I do my field work. So with that, I would like to pass it on to Justin.
Okay, thanks, Alex. So hi, everyone. I'm Justin Valliere. I was a Kretz postdoc fellow from 2016 to 2019. And like Alex, I worked with Phil Rundle at UCLA. And my practitioner mentor was Arena Irvine for the National Park Service in the Santa Monica Mountains. At the time, she's since moved on to a different office. And currently, I'm an assistant professor at California State University, Dominguez Hills, which is here in the south part of Los Angeles. So I consider myself an applied plant ecologist or a restoration ecologist. And the focus of my research when I was at UCLA and since then has been to really understand how will native plant communities respond to different drivers of environmental change. So climate change, drought, invasive species, air pollution, things like that. But more importantly, how can we develop really practical solutions for ecological restoration in the face of this change? So a lot of the science I do is maybe not the most glamorous, but aimed at understanding okay, how can we actually get plants in the ground that are going to survive in the face of environmental change? But to kind of echo what Elizabeth was saying with her talk earlier on, what I see as one of the most important steps that we can take to conserve biodiversity here in Southern California and really worldwide is to really make sure that we're training the next generation of conservation scientists. So that's really the whole reason I ended up pursuing a career in academia, which even when I was a postdoc, I didn't realize that's what I wanted to end up doing. But working with students has been the most enjoyable part of being a scientist for me. I think we also need to really recognize that this includes engaging students from diverse backgrounds and diverse communities in restoration research and conservation science, especially groups that have been or continue to be underrepresented and underserved in conservation fields and really science as a whole. So I think that's something that the Kretz Center is really well poised to do, and something that I tried to prioritize when I was a postdoc at UCLA. I should mention that one of the undergraduate students that worked with me in Phil's lab is actually going to be joining me next semester at Dominguez Hills as a master's student. So who knows, maybe she'll end up being one of the future Kretz fellows. And I think that would be a really great complete circle. Okay, so yeah, just to end, I'm really excited to continue doing research and restoration with students here in Southern California, potentially collaborating with the Kretz Center some more. And with that, I will hand things off to Luke.
Thank you so much, Justin. So I'm Luke Brown. I was a Kretz postdoc from 2017 to 2019, working with Victoria Sork at UCLA and also partners like Jessica Wright at the US Forest Service. I'm currently a postdoc at the Yale School of Environment. So I'm out on the East Coast right now and the sun is going down. So I apologize if it's a little dark. In terms of thinking about the biggest issues in conservation, I mean, there's so many to choose, but I'm primarily a forest ecologist. I think a lot about trees and I work a lot with trees. And you'll have probably heard of this push towards reforestation and planting trees as sort of a natural climate solution, where if we're able to plant enough trees, there's potential to, sequester carbon out of the atmosphere and help countries and help individuals reach sort of net zero goals that we have in order to stop climate change. So I'm all for that, and I think it's amazing. And there's these really big, impressive efforts to say plant 1 trillion trees over the next couple decades. And one thing that really interests me as a conservationist and as a scientist is, well, how do we do this, and how do we do this well? And can we do this based on a scientific approach rather than just going out into a forest and just grabbing seeds at random and planting them? But is there a way that we can do it that we can really enhance the potential success of these different initiatives? Because it's a lot of work to plant all these trees, so we want to make sure that they succeed. And so as a Kretz postdoc, one of the project that I was working on is how can we sort of take advantage of modern genomic techniques to give us insight into which trees are going to be the best sources of seeds for us going into the future. Because the environment that the trees are experiencing now is going to drastically change over the next couple of decades. So a tree that grows well here might not grow well in that same spot in 20, 30, 50 years. So what we did is we used genomic techniques to sort of gain insight into what individual genotypes do well under different future predicted climate scenarios, so that we can develop this approach where practitioners and conservationists can go out to the field, genotype a tree, and be able to determine with some level of certainty whether or not that individual is going to produce offspring that will do well under different climate scenarios as things get warmer, as things get drier, et cetera. And so that work was published right before the pandemic, and I'm hopeful that it will have an impact. It's been making its way into policy briefs and influencing policy a little bit like that, but I think the sort of wave of the future for conservation is integrating a lot of these genomic techniques. And so hopefully what I accomplished with colleagues at UCLA and through the Kretz Center will sort of build a foundation for what is possible going forward to help ensure that these huge initiatives for planting trees can be successful. So with that, I'll pass it on to Samantha.
All right, thank you, Luke. Hi, everyone. I'm really excited to be here today. I'm Samantha Catella. I'm currently a postdoc at the University of Pittsburgh, and I'm talking to you from Pittsburgh right now. And I'm gonna start my Kretz postdoc this fall, which I'm really excited about. So a major problem that I see in biodiversity conservation is that once biodiversity declines in an area, it is really difficult to bring it back. And so my research goals revolve around figuring out how to bring biodiversity back in restoration projects. And to do this, I work with land managers to identify what barriers they're facing to restoration, like why are these restoration projects failing, or why are they so expensive or so difficult to upkeep? and then together figure out how to overcome those barriers. So right now in my current postdoc, I've been working with New York State Parks and the Western New York Western New York Land Conservancy to implement a restoration experiment along the Niagara Gorge. And the problem they face there, that many land managers face, is that they can manage invasive species within that area. But because invasive species are so prolific and so common, that even when they control invasion in a certain area to increase biodiversity, because invaders often form monocultures, what they're seeing is that they get reseeded from outside areas. And so this thistle invasion just continues after they try to plant natives. And the thistle are great competitors, they take over, and it's this real problem that they have. And so my work there is investigating a process called biotic resistance. And biotic resistance is this observation that species-rich communities are less invaded than species-poor communities. But how that occurs, how biotic resistance happen, it's not well understood. And so my goal with that project is we're going to figure out how biotic resistance works by testing a number of different hypotheses. And then we're going to translate that knowledge into future plantings so that those plantings will better reinvasion. And sneak preview from that, I just collected data, and it looks like spatial arrangement of those native species that the restoration practitioners are putting in is really important. And as an incoming Kretz fellow, similar to this past postdoc that I've been doing, I asked the Nature Conservancy, I'll be working with Nathan Kraft at UCLA and Sophie Parker at the Nature Conservancy, and I wanted to know what problems they currently face in their work in LA. And a key issue that they need resolved is to figure out how to increase success and resilience of urban plantings for stormwater projects. And restoration is always hard, but restoration in a city is extra hard because plants face so many more stressors than they do in rural areas, including extra pollution, higher temperatures, increased abundance of invasive species, etc. However, our native species do persist in urban environments. And so we hypothesize that these plants that are persisting in the urban environment may be more successful in restoration projects than seed sourced from rural areas, because they may be locally adapted to those harsh urban conditions already. So my work in LA to test this hypothesis, and what we find is going to help organizations like the Nature Conservancy and California Gardens and Seed LA and these other organizations that already collect seeds for restoration projects. It's going to help them determine where to source their seeds for future restoration projects. So thanks for listening. I will hand this off to Yosha.
Thanks, Samantha. Hello, everyone. My name is JiaBin Huang. I joined Brad's lab as a Kretz postdoc in 2018, and that's where I still am here at UCLA. 2 weeks ago, the City Nature Challenge, which is a huge community science initiative, took place over a period of just 4 days, and more than 2,600 species were found here in Los Angeles. Now, I have nothing to do with the City Nature Challenge, but my research has everything to do with protecting nature in this city. And I asked myself, how do species navigate urban areas? What about a city allows species to move around? And what prevents this movement? So picture a lizard or a butterfly or a spider, if you don't mind, in your backyard. Does it ever make it across the road into the next urban green space? What about freeways and traffic? Or more generally, how connected are populations in a city? So my research measures this by looking at the genes of species and quantifying similarities and differences between populations sampled at various different locations. So some of these populations may be separated by a freeway or residential areas, while other populations may be linked by corridors or urban green spaces. Now, by combining the genomic data with the data on the landscape, we can quantify the impact of the city's infrastructure on connectivity. And this is a vital piece of information as it allows to better inform conservation management. So for example, by identifying specific locations which provide the least connectivity, we now know where to direct future restoration efforts, increasing their overall efficiency. And I'm proud to say that the data set that we've assembled for Los Angeles is the largest data set of this kind to date. It includes lizards, butterflies, spiders, slugs, flies, plants, overall more than 5,000 individuals, which were all sampled across the LA Basin and its surrounding wildland areas, such as the Santa Monica Mountains or the St. Gabriels, together with our partners, the National Park Service, USGS, the museum, and the Nature Conservancy. So hopefully this data set will also allow us to deliver solutions for urban areas elsewhere. Globally, rates of urban land conversions are currently greatest in Southeast Asia and Africa. So with our data set, we can potentially help designing urban areas in a way that makes them more biodiverse and more enjoyable for all to live in. And that's it from the Los Angeles Genomics Project. And I turn over to Eric.
Hi, my name is Eric Abelson. I was a Kretz postdoctoral fellow from 2014 to 2015, and I'm currently at the University of Texas in Austin. When I think of the largest challenges for biodiversity conservation, I'm especially concerned with human-driven global environmental change. Globally, climate change and habitat loss pose tremendous threats to social and ecological integrity. And I think we have a challenge to develop a better understanding of organisms in current landscapes and how they respond to both the steady changes imposed by humans, for example, by burgeoning urban areas and increasingly dense road infrastructure, as well as increasing climate variability, for example, frequent extreme weather events. I think that one avenue for approaching this problem is the expansion of established long-term monitoring of natural systems, monitoring weather and ecological conditions, variability, and how organisms respond over many years. Many aspects of ecological research are not easily brought indoors and recreated in a laboratory, and we need data and observations from real-world monitoring and experiments, including observation of how systems respond and rebound from variability in environmental conditions. This is something that field stations, those protected natural areas set aside for research, are well poised to facilitate. These data are important to not only understand how wildlife respond in current landscapes, but we can also use insights gleaned to better understand how wildlife respond in the future when these very rare events become more common. As just one example, historically, Los Angeles experienced an average of 4 extreme heat days per year. But by the end of the century, projections for the average number of extreme heat days per year may be as high as 30. And it's through collection of long-term data sets that I believe we can make progress on a number of fronts. For example, I collaborated with the National Park Service's Santa Monica Mountains National Recreation Area during my Kretz fellowship to examine a decade of mountain lion movement data in the Santa Monica Mountains. Coupling those data sets with other long-term research like regular plant surveys at UC field stations, we can begin to piece together what animals are doing in complicated landscapes during these extreme events. Collecting long-term data is not at all new. In fact, some of the most impactful science has come from long-term data sets. But the pace and scale of environmental change is new, and we need new approaches to expand how we conduct these types of research avenues. Building on work that I began during my time at UCLA, in my current position at UT Austin, I'm working on a project to set up long-term automated monitoring at field stations with the goal of capturing every sound and every sighting of wildlife on the landscape using remote sensors, and then coupling that with artificial intelligence to do the heavy lifting of identifying each of those sights and sounds so that ultimately we can identify large-scale ecological patterns. The ability, for example, to examine soil moisture, insect activity, migratory bird timing, mammal abundance can facilitate insights into how changing climates may be disrupting connections between many different species. One area of need and an area of my interest in general is to leverage increasingly affordable monitoring equipment coupled with advances in artificial intelligence to explore the promise of expanding our abilities to understand patterns in nature in unprecedented ways. And with that, I have the privilege to hand it off to my colleague, who I have tremendous respect for, Rachel Blakey. Thank you.
Thank you so much, Eric, for that lovely introduction. I'm Rachel. I'm a current Kretz postdoc. I'm in my second year, and I'm partnering with Seth Riley and Jeff Sikich from the National Park Service and Dan Blumstein from UCLA. So I'm originally Australian, if you haven't guessed. I received my PhD from University of New South Wales. And since that time, I've been working mostly in applied research, addressing the relationships between wildlife and vegetation structure, most often in the context of forest management and fire ecology. So the biodiversity problem I want to talk to you about today is a big problem both in my former home and also my current home of 7 years in California, and that's wildfire. So in both of those locations, our ecosystems have evolved with fire and are adapted to fire. However, we are seeing increases in the severity and the incidence of wildfires over time in these fire-prone regions. And that's due to climate change, increasing droughts and fire weather, fire suppression activities, human ignition, and a number of other things. So what I'm really interested in, what I see as a big interesting problem to try and solve, is how do we promote wildlife resilience to wildfire in landscapes that are also shared with humans? So I wanted to tell you a little bit about my current postdoc. So we're working on one of our top predators in California, which is the mountain lion, as Eric has also worked on. And we have this amazing opportunity to work on the state's most urbanized population of mountain lions here in LA, in Santa Monica Mountains, who also had about 50% of their hemmed in, highly urbanized habitat area pretty much decimated and flattened by the Woolsey fire in 2018. So because they are territorial and they're ambush predators, this has led them into a kind of trade-off between avoiding humans and urbanized areas with avoiding each other, potentially adult males who can get quite aggressive. So we've been investigating the risk-taking behavior of these animals in that context. And also we're going to be augmenting that work by looking at the 3D structure of vegetation, both before and after the Woolsey fire, to see if we can identify whether these animals are exhibiting resistance to fire, so using the same habitats, and oh, sorry, I thought I saw a note, using the same habitats after fire, or whether they're showing flexibility, so they might be changing their hunting strategies. And finally, the other area where we're lucky enough to be investigating in this postdoc is we're collaborating with a number of statewide mountain lion biologists so that we can get at the different characteristics of pre-fire vegetation and also burning characteristics like configuration and burn severity and how that influences mountain lion use of post-fire landscapes. And finally, one of the things that I really value and enjoy about the Kretz postdoc is that it has opened up a broader reach for my science. So I've been able to collaborate with and work with mountain lion biologists across the state, as well as stakeholders and managers through government meetings. I've been able to mentor a number of students, both in the National Park Service and UCLA, as well as being in discussion with the Natural History Museum of LA about increasing outreach about our LA mountain lions. So with that, thank you very much. I'm going to introduce my colleague, John Benson.
Just to tell you, John is recorded, so hopefully we'll get his sound sorted out in just 1 second. Gary, let's see, give it one more try and then maybe we'll just move on.
Hi, everyone. I'm John Benson. I was a Kretz postdoc from 2014 to 2016, and I'm now an assistant professor of vertebrate ecology at the University of Nebraska in Lincoln, Nebraska. So the problem I work on for conservation is the general problem of the extinction of species due to human stressors. And the more specific problem is that many large carnivore populations, and I study mostly large mammalian carnivores, many of these large carnivore populations are existing and persisting often in human-dominated landscapes in small isolated populations. And so my strategy is to study these small isolated populations. And I have to admit, as much as it's a serious conservation problem, I'm fascinated by the dynamics of these small populations, and particularly the interplay between demography and genetics. So I study the dynamics of these populations. I construct very realistic population models to model those dynamics. And within those models, we can test out different management strategies. In some cases, that has to do with harvest. In some cases, that has to do with how do we get more immigration and gene flow into these populations. So the first example I want to share with you is the work that I started as a postdoc with the Kretz Center, working with Seth Riley and Jeff Sikich of the National Park Service, studying the mountain lions and the isolated small population of mountain lions in the Santa Monica Mountains that I'm sure you're all familiar with. And what we did was all the great data that Seth and Jeff had collected over the years, we put that together into a population viability analysis. It was an individual-based model, and we were able to explicitly link demographic processes, so survival, reproduction, and population growth, and genetic processes like inbreeding and the loss of genetic diversity. And we projected the model forward 50 years, and we were able to make predictions about what might happen in this population. The population dynamics were generally pretty stable, although in such a small population, there was still the probability of extinction due purely to demographic processes. So in other words, a bad couple of years of survival or reproduction. But the bigger problem appeared to be that genetic diversity was declining at an alarming rate. And so our model predicted that within 50 years, you might get to the point where a much higher probability of extinction could occur. And so then we also modeled different scenarios involving additional immigration into the population, immigration that might be possible by way of a highway crossing structure, which, as you may know, is being planned over the 101 freeway to allow more mountain lions and more gene flow into this population. And so our model, I think, really provided the scientific basis and suggested that, yes, with modest increases in gene flow and mountain lion immigration, this population could persist into the long term. I don't want to take all night. I'm actually sorry I couldn't be with you live. I'm down in South Texas meeting with folks from the Cesar Kleberg Wildlife Research Institute that study the highly endangered ocelot. And they've got me out running around in the field down in ocelot country all day, so I couldn't be with you. But I'll just say that some other things that my lab is working on at the University of Nebraska. We are actually now studying, a PhD student and I are now studying mountain lion ecology in collaboration with CDFW and Justin Dellinger across the state of California, collaborating with a bunch of great researchers all over California. I'm also studying elk and mule deer in Nebraska. And I'm in the early days of helping to do research in a full carnivore guild reintroduction in Mozambique. So we hope to do some great things in terms of research and conservation in Southern Africa with that project. And with that, I'll just turn the mic over to Gary. I think you're next. Thanks a lot, everyone.
Yes, it's me, Gary Butcherelli, and I currently am a adjunct assistant professor at UCLA. Let's see, my postdoc was a 3-year collaboration with the National Park Service and Kretz Center to study local amphibians that breed in streams in the Santa Monica Mountains National Recreation Area, which, if you didn't know, is the largest urban park in North America. The focal amphibians were two species of tree frogs and the California newt. And a little bit of natural history. A newt is a type of salamander, just like a toad is a type of frog. The goal was to understand the genetic health of these three amphibians so that the National Park Service and other land managers like California State Parks and local land trust agencies can understand and really know which species and streams where amphibians breed are in need of the greatest protection or urgent conservation help. And that may be because individuals at a stream have low genetic diversity because of excessive inbreeding, or maybe it's net positive. In effect, each species at every stream is actually in good genetic health. And so there's really no need to prioritize their conservation. Well, based on the results of this work and ongoing research I do in the Santa Monica Mountains and throughout Southern California, our stream breeding amphibians are not doing well at all. Obviously, rain is kind of important, and unfortunately, we're in a drought and doesn't seem like that's going to bust anytime soon. And there are actually many other factors contributing to their problems that they're having. And I can talk about that more in the breakout room. But the real issue is there are all these negative effects that are leading to these streams being degraded to a point where newts and frogs are actually sick and dying. So today I want to take this opportunity to propose that streams in the Santa Monica Mountains National Recreation Area actually be granted individual legal status and associated protections through rights of nature law that defends waterways and species, and recognizes that species and ecosystems are not just resources for humans, but living entities with their own rights. So if anybody out there has a direct line to Richard Bloom or any of our other local legislators that would like to help with this and make it happen, send me a message. You can find me online or come see me in the breakout room. And with that, I will hand this over to my dear friend, Jesse.
Hey everyone, thank you, Gary, for that introduction. And I also wanted to just thank you all, attendees and everyone, for showing up tonight. We are all very passionate and excited about our work, and so it's a pleasure to have you all here to listen to us talk about it. My name is Jesse Grismer. I was a Kretz postdoc from 2016 to 2018, working with Brad Schaefer on basically using a large genomic data set to determine if the rubber boas that we have here living in our mountains of Southern California represent a distinct species and should be listed on the endangered species list. I am now an assistant professor at Valliere University here in Riverside, California, where a lot of the research in our lab that is done is on reptile and amphibian conservation in Central and Southeast Asia, but also here, a lot of it here in Southern California. And so my response to the prompt of what is one of the biggest challenges for conservation from my work with reptiles and amphibians, which happen to be really good bioindicators of the health of an ecosystem just due to their sensitivity to temperature and levels of moisture, is the shrinking and the reduction of natural habitat. I'm a Southern California. I grew up in Southern California. My father is also a local herpetologist, so I grew up chasing reptiles and amphibians in all these areas since I was 4 years old, all through high school, where we'd be out looking for horned lizards with your friends and getting up to stuff teenagers shouldn't, up in the hills and things like that. And moving back here after been away for 14, 15 years, I've just noticed that a lot of those places I used to go to are now a winery or have become a housing tract. And a statistic that I like to put out there that I feel really drives this home is, I got my PhD in the state of Kansas, which has a total population of 2.2 million people. The population of LA just passed 4.4 million people. So 2 times the entire state of Kansas's population just lives in LA. And as you heard from, Yosha previously, this large population is also coexisting with this massive amount of biodiversity. And so this creates interesting challenges in terms of management of, what chunks of habitat get saved, which ones get carved up into housing tracts or other developmental agencies and things like that. And so a lot of my previous work revolved around, exploratory research in parts of Southeast Asia, looking for looking for undiscovered new species of reptiles and amphibians and describing them to the general public and the scientific community. And so I brought a lot of those exploratory skills to my work while I was at the Kretz postdoc, and the work I've been doing in Southern California since then has been inspired by that time, is using that same kind of exploratory techniques of tracking down historical museum records, using analyses of habitat suitability to not only quantify like, hey, how much good habitat is left, but where haven't we looked for these animals? When you're dealing with threatened and endangered species, knowledge is basic distributional data, basically knowing where these animals occur is highly, highly valuable. And once you have that, we can gather tissue samples for them to construct these large data sets to say, all right, how are these populations connected? Who are they descended from? Or are they isolated and represent a distinct species? In the most extreme cases, represent federal funding on the endangered species list. And the great part about this is you don't need a degree to make a discovery. Anyone can go out and make a discovery. So this is a great opportunity for these city challenges through iNaturalist, citizen sciences, giving talks to local nature societies, giving nature walks, but also taking your herpetology or ecology class out on a hike. Anyone with an iPhone and an iNaturalist can start documenting these species because we can't conserve what we don't know exists. And with that, I will pass it over to Dave.
Thanks, Jesse. Man, you give really good Zoom talks, and now you've made me nervous because I have to follow you, but that's okay. Hi, everyone. My name's Dave Deversa. I am a current Kretz postdoc mentored by Brad Schaefer and Jamie Lloyd Smith and partnering with Rob Grasso from the National Park Service. And on the topic of biodiversity conservation, there's an issue I like to call the connectivity conundrum. For rare and vulnerable species on the brink of collapse, is it better to create these highly connected populations through popular interventions like movement corridors, or would it be better for them if we just created these very isolated groups to serve almost as refuges from stressors like disease? And this question actually just came up the other day during some work I was doing with the National Park Service. And in both academic and non-academic settings, it remains unresolved. This connectivity conundrum is a major problem for biodiversity conservation. And my Kretz postdoc project is trying to resolve this connectivity conundrum by studying endangered Yosemite toads in the Sierra Nevada range. This past week, the Yosemite, what are they called? The Sierra Aquatic Research team at Yosemite National Park and I were out capturing as many Yosemite toads as we could. And at the same time, some of the Kretz affiliates are collecting satellite imagery of the sites where toads used to breed. And these data will help us understand the risk that connectivity poses for outbreaks of a deadly fungal disease called chytridiomycosis. Some real insights, I think, are going to be provided by experimental introductions of Yosemite toads. So the San Francisco Zoo actually is breeding Yosemite toads in captivity and rearing them there so that we can eventually release them at various sites throughout Yosemite National Park, some of which are highly connected and some of which are more isolated. So we will get to see firsthand whether connectivity is beneficial or detrimental to this charismatic, but really threatened California endemic. Thank you.
Okay, thank you, Dave. Thank you, everybody. And that is our list. Those are the postdocs. And, you know, I've got a big smile on my face. Presumably, I hope you can understand that, because to my mind, this is amazing. This is a collection of people from beginning, fresh out of graduate school folks to relatively, I suppose, seasoned professionals, if you can call any of these young scientists seasoned, doing just phenomenally interesting, diverse, challenging work. They're all, one of the things I wanted to say about our postdocs is that to a person, they have all stayed in the conservation arena. Not one of them has said, you know, the heck with it, I just can't take this anymore, I'm getting out. Every one of them is still in. They may be in academics, they may be in the NGO world, they may be looking towards government positions, whatever. It doesn't matter, right? What matters is that they're committed, they're passionate, and they're making a difference. And an awful lot of them are doing so in California. Okay, what we're going to do for the next few minutes, and just a few, is try to answer some of the questions that have been submitted in the Q&A. We've got a bunch, so let's try to zip through them. The first one says, Zach, do you think that climate change and habitat fragmentation have larger effects here in LA than in other areas for these butterfly species that you study? Zach?
Yeah, thanks, Brad. The one-word answer is yes. As was mentioned by, I think, Jesse, like the population of Los Angeles alone is creeping past 4 million now. And so just considering the butterflies alone, I think you have 3 endangered species there, the El Segundo blue, the Pelos Verdes blue, and also the Kino checker spot, which is more of a Ventura County thing. And really, if you look at the threats to these, it's all habitat loss and fragmentation. So just the sheer abundance of population, particularly in this kind of sand dune habitat and coastal scrub, these habitats are really unique. So they contain a lot of what we might call like endemic subspecies that are only found on that particular habitat. And these are also the same kind of places that we want to live in. And so that's kind of a natural conflict, especially in a biodiverse state like California. And so that's the habitat loss angle of it, is just the sheer urban sprawl and not only the sand dunes, but the, particularly the coastal scrubland. That's a very popular ecosystem for development. So that's a huge threat. And then the climate change in Canada, sometimes even half of our news is consumed by the droughts that California faces, particularly in the south. So I gather that you guys are experiencing the most extreme drought since 1976 now. And so we're really feeling those effects now that climate change is particularly accentuated within the state there, with lower precipitation being the culprit. Butterflies are particularly affected because they're dependent on specific host plants that may not be that drought resistant. So it's not only the individual species themselves, but the interactions between these multiple species that are important to consider. So that's the long answer. The short answer is yes.
Okay, thank you, Zach. And everybody, all the panelists, just leave your cameras on. It's fun to see you. The next question is for Elizabeth, and I'm hoping everyone is still on. Why do you think there's been a decline in scientific literacy in the last decade? You know, and I suppose really particularly in the last decade.
Yeah, that's probably a little bit of a dangerous topic, but I think there's probably a couple of things. I think one is the internet. The internet does great things. It allows us to find each other and find wonderful, correct information, and it also makes it really easy to find nonsense, and it makes it really easy for people to propagate nonsense. And I think one good example of that is the Flat Earth Society. I remember hearing about the Flat Earth Society sometime in the mid-2000s, and I thought it was like a joke. And now, not only does it have like thousands and thousands of people involved in it, but we've got celebrities with large platforms trying to convince people that the earth is flat. And I think 30 years ago, there might have been a few dozen people around the world who thought the earth was flat, but they couldn't find each other. Now they can find each other and they can share that, and they've got platforms to talk about that. I also think that science has become politicized. I don't think that there's any way to avoid that as part of the conversation. I think that there were probably 2 topics that really that really illustrate that. One is climate change. There's been a lot of money by industries and politicians who've got a stake in claiming that climate change isn't real. And the other is the anti-vax movement. And also both of those things get traction on the internet as well. And then I think in the past year plus, we're also seeing the same thing happen with COVID denial. So I think that the politicalization of scientific literacy can go a long way towards explaining it. So I have some things that I think could help us with that, an emphasis on critical thinking in schools, an emphasis that funding agencies start to grapple with some of these questions, but that's where I land on that.
Okay, thank you. Thank you very much for that. It's, I don't know, those of us with a long perspective, and I certainly have a long one at this point. I'm not sure it's actually, I don't know, maybe it's worse than the last decade. Maybe it's a gradual decline. Anyhow, thank you very much for your answer to that. The next question is a general one. How do you hold on to hope, especially with regards to the ongoing mass extinction? And I'd like two people to answer. I'd like one from Alex, who is working in the tropics, and I'd like one from somebody else who's local, and that can be anyone who hasn't spoken yet. So Alex, why don't you start just briefly? How do you hold on to hope, especially with regards to the ongoing mass extinction?
It's definitely challenging to see mass extinction, and we focus a lot on climate change. And my lab specifically, we're really looking at tree mortality. So entire regions of forests. I have another audience member in the background right now. Sorry. Excuse me. So we're seeing, entire regions of forests, die out on every continent except for Antarctica. So that can be troubling. But I think what helps me hold on to hope is when I see the work that I'm doing and the work that my colleagues are doing. I really do. Maybe I'm just more of an optimist, but I think it does have an impact. And so I guess that's one way that I hold on to hope is, seeing the impact that my work has, that my colleagues have. The project that I'm on right now, we have over 100 scientists from different countries around the world. And it's this huge effort. Everybody's working towards this one goal. And I think we're going to achieve it. So when I see, yes, there's a lot of darkness and climate change, that's scary and terrifying. And I know there is science illiteracy. Some people, want to think it's a hoax, but I guess I have people in my family circle who used to think it's a hoax, and now they don't. And so I know that's an N of one, just me, but I think there is hope. So that's my perspective.
Right. Okay, and very briefly, somebody from California who hasn't spoken yet. I don't know. I'm looking at our panelists here. Jesse.
Go.
Jesse. Go.
It's a two answers here. One is the audience members. You guys all showed up to hear. We're in this because we love this. In some cases, it's our job, but the audience members showed up to hear about this and the importance of it and how it's going. So you all, number one, but also oddly, my niece. She's 6 years old and she loves going on Jeep rides with her grandpa in the desert. And the last time they were on a big Jeep ride out in the desert, she made him stop, and she got out and ran out into the desert and picked up a piece of trash and came back to the Jeep. And so I'd like to think that that's just probability alone would suggest that's not an isolated incident. There's probably other bunch of little girls out there running out of Jeeps to pick up garbage. And so I think we just need to teach the younger generation to care about living things and just to care about being kind and to be aware that their actions impact other living things.
Beautifully said. Thank you. Okay, next up, I'm going to skip one and go to someone who asks, Justin, you mentioned that it's important to include people from diverse backgrounds in conversations about conservation. I think that is an important starting point in promoting environmental justice. Are there ways that your research projects are addressing and promoting environmental justice?
Yeah, that's a really good question, I think. And I think it's like, it's not just like engaging people in conversations, it's engaging people like directly in the work itself. And the short and honest answer to that is maybe not as explicitly as I would like to do in terms of my research. It's mostly, I think, in terms of my community outreach and teaching that I am able or have been able to address that more explicitly. But I do try to find ways to incorporate that into my research. So there was this one study that I published when I was at UCLA that I started years before, looking at grassland restoration in Southern California. And all the data was collected for 5 years by 8th grade students from a Southern California school district. And some folks kind of scoffed at the idea that 8th grade students could collect publishable quality data, but they did. And they became like really adept scientists and ecologists and naturalists. So I think just like starting early is one great way to do that. And just as scientists recognizing that community scientists can play an important role in that process. And then just really briefly, I've been trying to be more intentional about that in my teaching. So I just started at Dominguez Hills this past year. And our student body is mostly students from South Los Angeles, and many of them just haven't been given the opportunity to think a lot about ecology, and that's on us, that's not on them. So giving them the opportunity to see that they are like the naturalists and ecologists of their environment, even if it's an urban environment. I gave a pre and post survey before I taught ecology this semester. And at the beginning of the semester, I asked students if they related to any scientists, and most students said no. One student said that she thought scientists were just white dudes with money, that that's who they were. But at the end of the semester, when I gave that survey again, it had changed. And I think part of that was, you know, like I'm a white dude, but I made sure to highlight work by diverse scientists. We explicitly talked about how class and race issues can influence urban environments and how that can influence wildlife, but also humans. So I think there are ways that we can tackle that with outreach, teaching, and research. Sorry, that was a really long-winded response.
That's great, thank you.
But I think we can all do better, including myself.
And certainly including myself. Thanks, Justin. I'm going to go to one quickly here. Sheldon Ellis asked a couple of questions. I'm just going to look at the first one. He asks, for genetic diversity, why can't mountain lions, so this is referring back to John Benson's taped talk, and other threatened animals be trapped and then released on the other side of the freeway in similar barriers? And the answer to that, I'm going to answer that because John is out studying ocelots in South Texas right now. But the answer is they can. You can set a trap and dart a lion and move it across the freeway and hope and pray that a lion on the side you moved it to doesn't kill it, or that it wants to be there and it doesn't try to go back. There's a number of things you can do. And I think if you work on reptiles and amphibians like I do, you can move some lizards and they'll probably stay there. If you want to move mountain lions, that's a different, that's a tougher lift, literally and figuratively. And I think the correct solution and the really beautiful solution is the overpass that is being actually planned. It was in yesterday's paper. Annenberg put up a $25 million funding, matching challenge for the overpass. It's already got something like $40 million ready for it, and it's going to happen. And the reason why it's going to happen is because John Benson did that study. I am convinced of that. His work was absolutely critical in showing that in 30 years, we won't have lions unless somehow either we dart them and helicopter them across the freeway, or we make a bridge so that they can walk across and do it naturally, one way or the other. It was John's work that did that. And it's to me, it's pretty inspirational. and something I'm very proud of as you know, a Kretz postdoc managed to make that contribution. Okay, we're going to do a couple of more questions here. And here's one for Professor Butcherelli for Gary. Have you observed any ways the local newt population has compensated or adapted to the drier weather as a result of the California drought?
I saw that question from Alec, one of my former students. Thank you, Alec, for the question. The answer really is it's a very complex situation that's unfolding in Southern California, where we've had this extended period of very dry conditions. And one thing we've noticed is that breeding newts, they've actually lost a significant amount of mass. And so the way we measure that is with a metric called body condition index, which is simply a ratio of mass to length. And what we observed in as little as 8 years was a near 20% on average reduction to body condition in our Southern California populations. And that's pretty alarming. We're seeing extremely emaciated individuals coming to streams that have very little water in sites where there would normally be very deep pools in streams where they breed. The Woolsey fire contributed a large amount of sediment through erosion with the loss of the plants along the mountainside. When it rained, all of that soil went into these streams, created channels. There's just very little habitat now. And so there's really subpar habitat. And we're also seeing very severe signs of some emergent disease. We don't know what it is. Brad and I and other collaborators with the National Park Service and the US Geological Survey have worked with international teams of researchers to try and figure out what's going on, and it's still unresolved. And this is extremely alarming for a couple of reasons, but most importantly because these populations are already on borrowed time, basically. Our data indicate that in possibly 3 to 5 generations, we might not even have newts in the Santa Monica Mountains or Southern California. So it's a very concerning time. And I think going back to this idea of hope, the hope is that we can address these problems and create suitable habitat or some sort of alternative that will allow species to persist even through climate change and all these complex stressors acting simultaneously on populations.
Very good. And, you know, I just want to say Gary has been a real leader in this area through his PhD work at UCLA and on through his postdoc, and now as an adjunct professor. He's worked closely with individuals from all of the relevant local government agencies and NGOs with Pepperdine University. So it's a team effort to try to kind of keep the Santa Monicas going, and Gary's been a real leader in that. Okay, we're going to do one more, and then if people are interested, we'll post in the chat the breakout rooms, and you can join those for 5 or 10 minutes or whatever you like, and chat with folks a little more. The last one I'd like to hear from is this. It says, for everyone, how does working with indigenous communities play into your work in conservation? And I'd particularly like to ask Luke if he could address that. That's Luke worked on oak trees, and oaks are a big part of the culture, the diet, the history of indigenous communities in Southern California. And I know it's something you've thought about a lot, Luke, so I wonder if you could perhaps field that one.
Yeah, sure. It's a great question. And similar to Justin's answers, the answer is we could all be doing more. We need to be doing more. We have an ethical obligation as scientists to engage with these communities in the areas that we're working on, especially the native populations. So yeah, one really fascinating thing about working with oaks in California is this really long tradition of native communities in California having just really strong cultural and cultural connections to the oaks themselves that continue to this day. And so working with Victoria, who has been a real proponent of building these bridges between the indigenous communities in Southern California and UCLA in particular, we're very fortunate enough to collaborate with some members of the Chumash community in Southern California, traditionally from the Santa Barbara area. But they have a really great cultural center in the Santa Monica Mountains, the Wistoyo Village. If you're looking for a place to learn more about this, that's a really excellent resource. And if you're looking for a cause to support, I'd really suggest checking them out. It's called the Wistoyo Village. And they lead a really great California naturalist certification program where you can take these courses and become a certified naturalist in California, but they do it in a way that integrates traditional ecological knowledge and indigenous knowledge. So half the program that they run is continuing on the traditions of how they, medicinal herbs, their connections to oak trees and things like that, but then also learning about sort of the more Western scientific natural history side of things. And so we were very fortunate to be able to join their group and sort of share our knowledge of what we know about the impacts of climate change on oak trees and what might be happening in the future. And then also there's this reciprocal exchange of their knowledge and their stories and their connections with oak trees. And I'd encourage, we all need to be doing more of that, but it's difficult. It takes a long time to build those relationships. And there's, as everyone knows, a very long history of really terrible things happening to these communities that makes it challenging to build those relationships.
Fair enough. Okay, thank you. I think we've still got lots more questions and they keep piling in, which of course is absolutely wonderful. But what I think we'd like to try at this point, since it is getting late and it is Saturday night, is to fire up, send out in the chat a Zoom link, and hopefully Gary or Colin can help us with that. And if you'd like to join one of the a chat room, one will be Elizabeth and Zach, one will be Alex, Justin, Luke, and Samantha, one will be Yosha, Eric, and Rachel, and one will be Gary, Jesse, and Dave. And there's some fascinating questions here. Join a chat room and ask them for a few minutes. We don't want to keep anybody for too long, but if you'd like to do that, it would be great. And the final thing I will say before we go into those chats, and I'll join one of them, I'm not sure which, is if you like this program, if you think it's a great program, if you think these are great people, I sure as heck do. And if you think they're worthwhile, we have funds to hire a postdoc a year for 2 years into the foreseeable future. I'd love to hire 2. I'd love to hire 5 because they're all worth it and they're all great. And if you have any capacity and you'd like to give us a hand with that, get in touch with me, go to the Kretz website and think about helping out, because that's how we make it work. We have our foundational funding, and then we put together other funding to help bring on other individuals and help keep these people in our community doing great conservation work that makes a difference.